Bismillah ar-Rahman ar-Rahim. Hello everyone, this is solving past paper 2020 October November variant 3-4 question 2. Now you are provided with the prepared slide from the examiner. It's labeled by M1. Now after you place the slide under the microscope, you will see this field of view, which is basically a transverse section of a stem. Now the first question in this paper is to make an outline for a quarter of this stem. Now you need to think about the different areas in this stem. You need to think about the different layers, and as we say always, different layers can be determined by basically the shape and the size of different cells viewed in this transverse section. Okay, try do your drawing, and then I'm going to show you how to make the outline for this for a quarter of this transverse section. Okay, now after you had your chance in drawing the outline for this slide, I'm going to tell you now what are the different areas or different tissues that you can identify in this slide. Now, as usual, guys, you can start with the epidermal cells, which is basically epidermal layer. Now that you can see at the very beginning of or the very outer layer of this transverse section. Now, if you look carefully, you will see that there are some detached part of the epidermal cells. Now, my advice is to ignore that. Now, during preparation, detaching or damaging for tissues might happen. And some slides in the exam, some slides would have this, other slides would not have that. So avoid drawing those detached areas and assume, okay, Ahmed, assume that those areas or ignore them basically. You ignore them and draw the epidermal as one connected line. So the first line right here is the epidermis. So I'm gonna show you how to draw the epidermis on the board. Yeah. So this is the epidermis, which is basically the very beginning of the outer layer of the transverse section. So again, notice that I ignored this part right here, and I acted as if it is not there. Now, the second layer that you can notice is this area right here. Now, this area is characterized by some type of tissues that is different than the epidermal cells. So basically, you can draw a line right here, which shows that you recognize that there is difference between those cells and those cells. So by drawing this line, you are making a difference or a border between those two different areas. Let me make it a little bit curved. So this is how it looks like. After that, if you look carefully, you will see that underneath the epidermal cells, there is a layer of cells that is different from the epidermal cells also. And this layer of cells ends right here, which is the layer of endodermis. The layer of endodermis basically is connected all the way from this, or let's say it passes through those structures. Now, if you are familiar with the dicot transverse section, or transverse section of dicot root, that would help you in understanding this slide, by the way. And this line right here, which is basically the endodermis. Remember that we are drawing only 1/4 of the specimen. Now, you might notice that this area right here, which is a vascular bundle, has two types of tissues. It has this one, which is characterized by large circles and thick walled, and this one right here, which is a little bit different, or it's actually different than the lower part. It is characterized by two different types of tissue. Now, I think it's very easy to guess that this layer right here is basically the xylem tissue vessels, or the xylem tissue which has the xylem vessels right here, because xylem vessels all the time is characterized by large circles, large lumen, and thick walls. Okay, now if this is the epidermis and this is the endodermis, as I said, there are some vascular bundles located here and there, so it's better to draw them. So here's two on the left, two on the right. Those are the vascular bundles that locates right here. Endodermis all the way to the middle of the structure is basically called the peth. So the peth is all this distance right here, starting from the distance from the endodermis all the way to the center of the specimen is called the peth. Okay. Now, again, from the endodermis all the way to the center of the specimen is called the peth. So the peth is this area, all of that area. And you can see that in the peth there are parenchyma cells. Now those cells are parenchyma cells. You are not supposed to draw cells, so you can't, let's say, draw cells in the outline. This is an outline drawing, so you cannot draw individual cells. But what you need to draw is this layer right here, which represents the end of the parenchyma cells. You draw it by one single line, which represents the end of that area. Now in the middle, in the center of that, let's say, peth, there is an air space. There is a lumen right here. So this part right here is basically a lumen, and you cannot draw a lumen, it's only an air space. So this is how the drawing gonna look like. To the whiteboard so I can show how the drawing. So this is basically how your drawing gonna look like. It is the quarter of the specimen. Okay, you can fix it from here. Now those feathery lines are basically should be fixed. So remember that I am drawing on the board using marker, which is a bit different than using a pencil. Okay? Okay, now the second part of the question is asking you to draw 4 touching cells or 4 cells from the central part of the cell. Now, what does he mean by the central part of the cell? Now again, if you look at the transverse section, you will see that this is the epidermis, this is the endodermis. Now, if you get from the endodermis to the central part of the cell, that area basically is called the peth. Now, we agreed earlier that this area is basically a lumen, which is an air space. The central part of the cell means you need to pick 4 cells of this area right here, which is basically parenchyma cells, that has this shape, just like, you know, it's kind of pentagons, it's sometimes hexagons, sometimes it is more into circle. So basically you need to pick four cells that touches each other. Now every one of those 2 cells touching 2 other cells. Now let's pick for example, let's pick those 4 cells. Let's say this is 1, 2, 3, and 4 cells. How would you draw those cells? So again, how would you draw those 4 cells? Basically, once you look at them, you need to consider that there are basically no air spaces between them. I think if you use your microscope, which is going to be much more clear and it's going to have more resolution, I think some of our students say that there is air spaces. Is there any air space between them, Ahmed?
Yes.
So Ahmed says there are air spaces between them. So I can't see them, but if you see air spaces, simply you draw them. Let me show you how you draw those 4 cells. So this is cell number 1. This is cell number 1, which is gonna look like this, something like this. Now remember, guys, that whenever you draw plant cells, you need to make two lines to represent the cell wall. So I'm gonna start with the first line, and then I'm gonna draw the second line after I draw the first one. So this is cell number 1, okay? It is something like that. And after that, I'm gonna draw cell number 2, which is looking like this. This is cell number 2. Now, if you look at cell number 4, which is something like this. So this is how we draw cell number 4. So make sure that cell number 1 touches number 2 as well as touch number 4. Number 4 touches 2 and 1, as you can see in the real view. This is number 3, I think. And that's it. Those are the 4 cells. Now you need to make 2 lines to represent the cell wall. So here's the cell wall represented by 2 lines. And make sure that whenever there is 2 cells touching each other, there is going to be 3 lines in common. So here is 1, 2, and 3 lines. And here is 1 line inside.
هون في air space يمكن.
Is there air space?
بالbig microscope.
In the microscope, there's big space or air space. Now, if there is an air space, simply you leave it. But if you can't see it, you simply don't leave it, or you make them without air spaces. Now, if you are going to label the cell wall is going to be the distance between the outer wall and the inner wall to represent the thickness of the cell wall. I think it's a very easy question and you can get full mark. Okay, the next part of the question asks you about making some measurement and some calculation. The question asks you to basically to calculate the mean length for the outer layer and the mean length for the inner layer, and then to find the actual length for the outer layer and the actual length for the inner layer. So how to do that? Basically, guys, if you use your ruler and measure the lines, which is A, B, and C, from this point, this is for line A, from this point all the way until this point, which is basically the beginning to the end of the transverse section for line A, as well as for line B and for line C, and you add them up, divide by 3. By that you will get the diameter, the average or the mean diameter for this structure. That's for the outer layer. Now, if this is the actual, if this is basically the measured mean diameter, how to find out the actual mean diameter? Now, if you notice here, right here in the question, he says that the magnification is 32 times. That means this image is magnified 32 times compared to the actual size. What you need to do is to divide the mean length that you have calculated on 32 to find out the actual length. Let me show you my calculation. So for line A it was 9.8 centimeters. You add it up to 8.9 for B and 9.7 for C. You divide by 3 to get 9.5 centimeters. And in order to find the actual length, what you need to do is to divide it by 32, and then you will get the answer of 0.296. Now, you need to make your answer accurate. As long as we are using centimeters, 1 decimal place is enough. So because your ruler can take only 1 millimeter, so that's going to be 0.2 centimeters. Okay, I'm using my left hand. 0.2 centimeters for the mean actual length for the outer line. For the inner line, you're going to do the same, but you are going to measure the inner line from this point right here to this point right here. How did I know that? Because basically the examiner gave you this information telling you that this is the inner line, and the inner line goes from this line right here all the way to the opposite side. You make the measurement for the 3 lines at the green marks as it shows board, and then calculate the mean length for the inner line, and then calculate the mean actual length by dividing it over 32. Again, 1 decimal place is enough for the answer, so the answer is going to be 0.1 centimeter because you need to follow the accuracy of your ruler. One more question remains in this paper. So here is a quick correction. This one would be 0.3, not 0.2. And also this one is 0.2. Okay? Now for the last branch of this question, you need to make a ratio between the mean actual length. If you read the question, you will see that the examiner asks you to make a ratio. You're going to make the mean actual length for the outer layer and the inner layer. It's better to write outer and inner. So right here, you're going to say the outer layer to inner layer. Okay. So 0.3 centimeter to 0.2 centimeter, you divide it by 0.2, and then you're going to get the answer 1 to 1.5. If you round it to a whole number, it's going to be 2 to 1. Okay, this is how you make the ratio. Okay, for the last part of this question, which is basically the comparison between the M1 slide, which is right here, and the figure 2.3, which is right here. You need to distinguish between those two specimens. You need to fill in the table that is provided for you, to fill in the feature and what is the difference between those two slides or two specimens regarding that feature that you picked. The first feature, guys, is the shape of the entire transverse section. If you look carefully, the shape of this figure 2.3 is basically more into circular, while this one is more into square. So if you write down the first feature, it's going to be the feature or the shape of the structure. It can be circular, and the second one is square. Okay, so make sure, guys, once you fill the table, to use the ruler in order to make a ruler to make rows, okay, to get the entire mark. Here's one more difference. Ahmed, he suggests that this one has an airspace inside, while the other one has no airspace inside. This is a very important difference between them. So what you're going to write the feature? The feature is airspace. You're going to say present and absent. Yeah. So the feature is airspace, or the airspace in the path. It's better than writing path. Both of them has path. The path is basically the area from the endodermis all the way to the center. So if there is airspace in the path, there is airspace right here, but there is no airspace right there. Another one. What do you think? The thickness of the cortex. I think they have almost the same thickness of the cortex.
Vascular bundles.
Vascular bundles, yes. What are you going to say about vascular bundles? Ahmed suggests that the vascular bundles in M1 slide is fewer, yes, less, but the vascular bundles in figure 2.3 are much more compared to the vascular bundle in the M1 slide. The feature would be the number of vascular bundle, less more. The feature, the shape of the structure, circular, square. And another feature is the presence of airspace, present, absent. Is there any other differences, Shabab, you can see? I think if you look carefully here into the epidermis of figure 2.3, you're going to find that there is much thicker epidermis, yes, compared to the epidermis right here in the M1 slide. Even though M1 slide the epidermal layer is made of two lines or two lines of cells or two layers of cells. Here is a thicker epidermal layer compared to M1 slide. Okay, this is the end. Thank you very much.